Hello and welcome to Sam Biology class. Friends, today we shall discuss about linkage. All organisms have large number of genes. The number of genes is much more than the number of chromosomes. We know that genes are located in chromosomes. So each chromosome contains a large number of genes. The genes that are located in the same chromosome and are transmitted together in the offspring are called as linked genes. The phenomenon of inheritance of linked genes together so as to retain parental combination in the offspring is known as linkage. The characters that are controlled by the linked genes are known as linked characters. In this figure you can see that this is a chromosome and these are the genes which are located in it. Because all the genes are present in the same chromosome, so there is a tendency for all those genes to be transmitted together. This tendency of the gene which are located on the same chromosome to be transmitted together is known as linkage. And the genes which exhibit linkage are called as linked genes. And the characters which are controlled by linked genes are called as linked characters. All those genes which are located in a single chromosome constitute a linkage group. In an organism, the total number of linkage group corresponds to the haploid number of chromosomes or to the number of chromosome pairs. Because human being has 23 pairs of chromosomes, so there will be 23 linkage groups. Whereas sweet pea has 7 linkage groups, and in Drosophila melanogaster the total number of linkage groups will be 4. Linkage was first reported by W. Bateson and R. C. Punnet in sweet peas, Lathyrus odoratus. These researchers crossed varieties of sweet peas that differed in 2 traits. One trait is color of the flower and the other trait is length of the pollen grain. The alleles of the flower color gene are capital R for red flowers and small r for white flowers. The alleles of the pollen length gene are capital L for long pollen grains and small l for short pollens. Capital R and capital L are dominant alleles. Pea plants with red flowers and long pollen grains were crossed to plants having white flowers and short pollens. All the F1 plants were found to have red flowers and long pollen grains, indicating that the alleles controlling these 2 phenotypes are dominant. After that, when F1 plants were self-fertilized, Bateson and Punnet found a peculiar distribution of phenotypes among F2 plants. Instead of 9:3:3:1 ratio, which is expected for 2 independently assorting genes, Bateson and Punnet obtained a ratio of 24.3:1.1:1:7.1. Among 803 F2 plants that were examined, the parental classes were significantly overrepresented, whereas non-parental classes were underrepresented significantly. Parental classes included 2 groups of plant. One group which has red flowers and long pollen grains, and the other group which has white flower and short pollens. These 2 groups together constitute the parental class. Remaining 2 groups, one group has red flowers and short pollens and the other group has white flowers and long pollen. These two groups constitute the non-parental class. Red flowers with long pollen grains and white flowers with short pollen grains are parental combination of characters, whereas red flowers with short pollen grains and white flowers with long pollens are non-parental combination of characters. Out of 803 F2 plants examined, 753 plants are in the parental class, whereas remaining very few, 50 plants are in the non-parental class. So plants that are included in the parental class are much more in number as compared to the plants of the non-parental class. What is the reason behind this? The reason is there is a lack of independent assortment between the genes which control the flower color and the gene which control length of the pollen grain. It is because of the lack of independent assortment between the gene for flower color and the gene for pollen length, parental classes were significantly overrepresented. Bateson and Punnet provided a complicated explanation for this lack of independent assortment, but their explanation was found to be wrong. The correct explanation is that the gene for flower color and the gene for length of the pollen grain are present on the same chromosome, so these two genes are linked. Because of the linkage between these two genes, parental classes were significantly overrepresented, whereas non-parental classes were significantly underrepresented. This figure provides a suitable explanation for this. In one group of plants, both the alleles are dominant, whereas in the other group, both the alleles are recessive. So this group of plants is homozygous dominant for both the alleles, whereas the other group is homozygous recessive for both the alleles. When these two groups of plants were crossed, all the F1 hybrids were found to have red flowers and long pollen grains. The F1 plants are doubly heterozygous. It is due to the lack of independent assortment between the genes for flower color and the genes for pollen length. We will expect the doubly heterozygous F1 plants to produce only two types of gametes. One type of gamete will have dominant alleles and the other type of gametes will have recessive alleles. In both of these gametes, the chromosomes are non-recombinant chromosomes, but it is due to once in a while crossing over between the two genes, alleles recombine and that result in the formation of recombinant gametes. However, the frequency of recombinant gametes depend on the frequency of crossing over between these two genes. Bateson and Punnet performed an intercross in F1. They could not perform a test cross. In a test cross, the offsprings could reveal the types of gametes that can be produced by the doubly heterozygous F1 hybrids. This figure shows the analysis of such a test cross. In this example, doubly heterozygous F1 hybrids having red flowers and long pollen grains were crossed to pea plants which are double recessive. Out of 1000 progenies that were examined, 450 plants had red flowers with long pollen grains and 470 plants had white flowers with short pollen grains. These two groups together, 920 plants had parental combination of characters, and remaining 42 plants had red flower with short pollen grains, whereas 38 plants had white flowers with long pollen grains. Together, 80 plants had non-parental combination of characters. These 80 plants were recombinants. In this example, the frequency of recombinants is equal to 80 by 80 plus 920 is equal to 0.08. Because it is a test cross, the frequency of recombinant gametes will also be 0.08. So, the parental combination of characters are more numerous than the non-parental combinations. Bateson and Punnet suggested that alleles coming from the same parent tend to enter the same gamete and are inherited together. This is known as genetic coupling. Whereas alleles coming from the different parents tend to enter the different gametes and are therefore inherited separately and independently. This is known as genetic repulsion. However, Bateson and Punnet could not provide a correct explanation for the coupling phase and repulsion phase.